So I believe that we finished with fats yesterday, with lipids yesterday, sorry. So we had 3 types of lipids. We had fats, phospholipids, and steroids or sterols. Today we're gonna pick up with proteins, and proteins is the biggest section of this chapter. And the reason is because, as I usually tell students, proteins do everything in cells. They are, they perform all the functions. Can you turn off your microphones? Can you mute yourself, please? Because you're being recorded. So proteins do everything in cells. So we want to be sure that we know a lot about proteins because we are going to spend a lot of time talking about them in the rest of the chapters. So this first slide that I've put up here, this is a list of all 20 amino acids. So proteins, the monomer for proteins, or the building block for proteins, is what is called an amino acid. And it is called that because, I'll give you a bigger picture here, because if we'll just look at some of them at this time. What I need you to notice and recognize for the monomer is that this amine group, carbon, and then a carboxyl group, and that's the words I'm using from chapter 4, from that table in chapter 4, that one panel that I asked you to take a look at and study. That's why they're called amino acids. So they're amino because they have an amine group. They're an acid because in some textbooks you'll actually see a hydrogen here, and in this case the hydrogen is not there because it's already been donated to the solution. Remember, acids are proton donors to the solution, so protons usually we express as a hydrogen with a positive charge. All of the 20 amino acids, they all have this same sort of backbone structure. They all have an amine group, carbon, and a carboxyl group. What is different about all 20 is each of the 20 have a different functional or side group, or you'll see them called R groups. And you can see they're in yellow here. They're all different. Some of them are rings, some of them are chains, and there are 20 amino acids because there are 20 different of these side chains. I'm just showing you the larger pictures. Some of them are positively charged, some of them are negatively charged, some of them are really big, as I mentioned, because they have rings, 1 or 2 rings. I'm not expecting you to memorize how to spell all of these, but you will need to be able to recognize an amino acid when you see one. And then later, when we get to like chapter 17, I believe it is, yeah, I think it's chapter 17, the amino acids will come back again when we translate RNA into protein. And then I will ask you to remember these words and that GLY stands for glycine. But you don't need to memorize that right now. What you need to focus on right now is that the monomer, the building block of amino acid, of proteins, is an amino acid. They get joined together in chains by, of course, guess what? A dehydration synthesis reaction, which we talked about yesterday, water coming out of the reaction, right? And this bond that they're highlighting in blue here, it has a special name. Unlike the other bonds that we would just say are covalent bonds or ionic bonds or hydrogen bonds, a bond between 2 amino acids is officially known as a peptide bond. Okay? And again, that's going to come back to haunt you in chapter 17 when we talk about how proteins are actually synthesized inside the cell. So in this case here, we have what we would call a tripeptide because there are 3 amino acids. We're going to build these amino acids into chains. When there are multiple amino acids in a chain, then we refer to it as a polypeptide. So let's take a look here. So here's a picture of a polypeptide. And notice that it gets a lot smaller, and we just get the 3-letter code now for each of those amino acids. Think of them like letters in the alphabet. You can put them together in almost, almost millions or billions of different combinations in order to make words. You can put amino acids in millions to billions of different combinations to make different proteins. And each of those proteins is going to have a different function inside the cell. So here's this chain of amino acids. For this particular protein, it is 125 amino acids long. They're usually numbered every 5 or more likely every 10. And again, what will happen when you see a protein that is spelled out in its amino acid chain, you don't see the whole amino acid word.
Okay.
You don't see the whole amino acid word, you just see the 3-letter code. Okay. Now, this we would call a polypeptide. It's a chain of amino acids. Remember, the monomer is the amino acid. That's the building block. There are 20 different building blocks, 20 different amino acids. This is the primary structure, notice, of this protein. It's called transthyretin. And transthyretin is actually a protein that makes spider webs. Okay, so here's the spider and its web, and we're looking at structurally what this looks like in a macroscopic version, meaning what we could see if we went outside and saw a spider web. This is the very, very microscopic version, right? We're seeing first the list of all the amino acids in this protein. And there are 125, 26, 27, looks like. Now, because most proteins are made up of more than one chain of amino acids, we refer to proteins having layers or levels of structure. There are 4 layers of or levels of protein structure, starting with primary structure, secondary structure, tertiary structure, and quaternary structure. And we're just going to run through those real quickly here. Primary structure is the list of the amino acids in order. Secondary structure, because those amino acids have those side chains, those R groups, right? And those R groups have different capacities for chemistry. Depending on the order of those amino acids, they don't just stay as a straight chain, okay? Those side groups cause chemical interactions that are going to fold and turn this molecule into what we would consider higher levels of structure or higher levels of organization. The primary structure is always the list of the amino acids in order. The secondary structure, there are 2 types of secondary structure. The first type we refer to as an alpha helix. That's this one. Okay, it looks like a telephone cord, or most of you are too young to have had corded telephones. Think about the spiral on the edge of your notebook. That's very similar to an alpha helix in that it's a perfect coil, and you can look down it and you can see it's hollow down the middle. Okay. That's one type of secondary structure. See, secondary structure called an alpha helix. Second type is called a beta pleated sheet, and this one isn't so as easy for me to show you an example of, but I would think of a beta pleated sheet. The molecule kind of goes up and down like hills and valleys, or think of like a broiler pan. It looks like that, actually. Most proteins are going to be a combination of these different types of secondary structure. Okay. Now, when the secondary structures, because we're either twisting that primary chain or we're putting it into this other shape, now it has the potential to further interact with other amino acids that in the primary chain might be quite far from each other, right? So amino acid 105 might actually, after the secondary structure gets formed, amino acid 105 might be quite close to amino acid 45, and maybe they do some chemistry with each other. That's tertiary structure. Okay, so this is our same protein. This is our same transthyretin protein, and this is what its tertiary structure looks like. So it has areas where it has alpha helix. These long lines with the arrows on them, those represent the beta pleated sheets. And these areas here in between, like these 2 beta pleated sheets, that just means they're not showing a particular secondary structure of either alpha helix or beta pleated sheet. This is an area where the primary chain of amino acids would still be visible. Another beta pleated sheet, sorry about that. Mostly beta pleated sheets, right? With just a little alpha helix. Now, For some proteins, this is it. They have primary, secondary, tertiary structure, and that's it. For many proteins, they have quaternary structure, which is multiple subunits or domains put together to become a functional protein. And in the case of transthyretin, which remember is spiderweb protein, it actually has 4 subunits. So the picture that I just showed you is this guy here. That was the tertiary structure. And you can see that there are 4 almost identical structures, polypeptides. This one is flipped and, you know, like almost a mirror image. And then you can see the 2 in the back. They colored them blue instead of purple. And they are also the same primary chain here, just that they're assembled into 4 subunits or 4 domains in order to be a functional protein. Again, for most proteins, they have at least tertiary structure, and many have quaternary structure. This panel is in here just to show you some of the chemical interactions that might happen between amino acids. The purple ribbon is supposed to represent the primary sequence of the amino acids, and they're showing you some amino acids interacting with each other that are far apart on that are normally maybe far apart numerically. Here's a picture of collagen. This would be a protein that has quaternary structure because there's 3 chains of polypeptide. See how there's one orange, one green, one blue. Each one of those chains is an alpha helix, and it's alpha helix only. Fibrous proteins that are like fibers or ropes usually have some version of chains of alpha helix and sometimes multiple alpha helix. Globular proteins that have a globby shape usually have a lot of beta plated sheet. Hemoglobin is usually our poster child for showing you protein structure. Similar to transthyretin, it has 4 subunits, 2 alpha subunits, 2 beta subunits, and then this area here is the iron portion where the oxygen actually binds. So each hemoglobin molecule can bind 4 molecules of oxygen. And they're showing you that inside a red blood cell, it is chock full of hemoglobin, right? And then the last panel for proteins is always this one to show you how important that primary sequence of amino acids is, because this is probably one of the most dramatic examples on the planet. Notice here, amino acid number 6 is supposed to be, I believe that's glutamine or glutamic acid. I'm not even sure because it's been a long time since I had to know those. Go back and look. What is GLU? Glutamic acid, right? GLU stands for glutamic acid. So if we look at hemoglobin here, normally amino acid 6 is glutamic acid. If that amino acid 6 has been changed by a mutation to the DNA and now it codes for valine, that is the cause of an abnormal shape to the beta subunit of hemoglobin, and the hemoglobin doesn't assemble in the same way as normal, and then it aggregates into this fiber that causes this sickle shape of the red blood cells, which diminishes how much oxygen can be carried by each hemoglobin molecule. So the reason we show you this is because we're trying to help you understand that the order of those amino acids is really, really important. And sometimes just one of them being changed causes a disease, right? Not always. This is, like I said, this is a rare situation, but it's a good example of what can happen if a single amino acid is incorrectly placed in the wrong place on that primary structure. Now, as far as the functions of proteins, as I mentioned, proteins sort of do everything in cells. I believe your textbook lists 8 functions of proteins. I would expect you to be able to recognize these functions. I'm not going to ask you for the examples. I'm expecting that you can recognize, for example, if I said which of the following is not a function of proteins, and I listed these 4 and energy, you would be able to say, oh, proteins don't provide, they're not the first choice for energy. We can store amino acids that can be then used for energy, but it's not like a carbohydrate, for example, which we would use for energy immediately. So here's 4 of those 8 functions. Enzymes for that whole section, we're thinking about chapter 1, energy and matter. Defensive proteins, storage, transport, we're going to see a lot of that in chapter 7. And then hormones. Actually, most of your hormones are not steroid hormones. Most of them are protein hormones. Receptors for chemical signaling between cells. So if you think about interactions, molecular interactions that we talked about on Monday, movement, and support. Okay, so those are the 8 functions of proteins. Okay, here's just some pictures of proteins because they're pretty to look at. So this is the space filling model. You might recognize this, looks kind of familiar to when we saw water trying to dissolve a covalently bonded molecule yesterday at the end of chapter 3. This is the ribbon model. This shows particularly highlights the secondary structure of the alpha helix and the beta pleated sheet. And then this would be more of a functional molecule where we can, where with a computer modeling program, these are the individual amino acids sticking out here in blue, and they're looking at a particular how bacteria binds to lysozyme, which is an enzyme that can break down the bacterial cell wall. So these sorts of models would be used particularly in pharmaceuticals, trying to determine where the interaction is and which amino acids are key for a protein's function. All these pictures are just the pictures from your book. So you have all these pictures. Someone just asked if I'm going to post these PowerPoints. Probably not for chapter 5 because we're just kind of going through them. I didn't select them out. You can just look at them in your textbook. They're all in there. I'm taking them directly from the book. All right, the last group of organic molecules, group number 4, is called nucleic acids. The building block of a nucleic acid is called a nucleotide here in the center. Let's see if I can make that a little bit larger. We can just look at this. If the molecule does not have a phosphate attached to it, then we call it a nucleoside. So if you've heard that word before, that's what it means. I'm going to use the word nucleotide. And I think in your note outline, I specifically stated each nucleotide has 3 parts: a phosphate group, a sugar, a monosaccharide, and a nitrogenous base. When the nucleotides are joined together to form a chain, that's when we call them a nucleic acid. No one uses this word, so you won't see that on a test. Polynucleotide. Okay. So let's go back and take a look at the different nitrogenous bases. There are 5 of them. Again, I don't expect you to memorize them. I'm not going to have this picture on the test and say, is this adenine, guanine, cytosine, thymine, or uracil? But I do expect you to be able to see this picture and say that's a purine. Okay, it's a nitrogenous base and it's a purine. How do we know? Purines, easiest way to remember, they have 2 nitrogen-containing rings joined by carbons that are double bonded together here. And the pyrimidines only have 1 ring, and there are 3 of those. To be totally honest, I always remember it. Purine, short word, big picture. So short word, purine has the 2 rings. Pyrimidine, which is long word, only has 1 ring. There are lots and lots of things that you're going to have to memorize in Bio 1 that are 2 things. And what I suggest to students all the time is just pick one of them to memorize so that when you're faced with it on an exam, it's really quick for your brain to either say, is that the thing I memorized or is it the opposite? And then make your choice. Okay, so like I said, for me, if I look at a picture of DNA, which I do a lot, I'm like, oh, purine permine. Okay, purine, big short word, big picture. Okay, yeah, adenine. Adenine's a purine. So for each nucleotide, you need one of these, a monosaccharide. And if it's DNA, that monosaccharide is going to be deoxyribose and a phosphate. The phosphate's always the same. If we take a look at the differences between deoxyribose and ribose, they are 5 carbon sugars. You'll notice they're numbered for you. 1 after the oxygen, clockwise, 1, 2, 3, 4, 5. Ribose, there's a hydroxide group on carbon number 2. Deoxyribose, there's a single hydrogen. Deoxy, missing an oxygen. When you put nucleotides that contain deoxyribose and one of these 4 bases, adenine, guanine, cytosine, or thymine together, you form a single chain. It's called a nucleic acid. And in DNA, those nucleic acids, oh, there's a picture of just the bases. In DNA, those nucleic acids are going to form a double helix. They are going to bond 2 chains. We're going to have 2 nucleic acid chains, and the bases we say are complementary. Adenine pairs with thymine, and cytosine pairs with guanine. A similar molecule that we call RNA for ribonucleic acid is most of the time a single chain. And instead of deoxyribose here, there would be the ribose instead. Unfortunately, here we go. Here's a picture of some RNA. Again, you may know this from somewhere in your past. The DNA is double-stranded in the nucleus. From the DNA, we're going to make a copy into RNA. So it's going to have that other sugar, the ribose, and a different base. It has uracil instead of thymine. And then this RNA is going to leave the nucleus, and it's going to be translated into proteins. So this process, we call this part transcription and this part translation. That is all in chapter 17, which we're going to get it later, right? So that comes a lot longer. That is the end of chapter 5. So I'm going to stop the recording now.